the one and only Sir Patrick of the Department of Biology. Welcome to Lecture 5. Now, we're going to talk about Cyanobacteria.
So, phylum Cyanobacteria, or also known as your Cyanobacteria, also known as your blue-green algae. The blue-green algae term is actually a misnomer because, the algae portion of that name, of the blue-green algae, gives it the impression of it being an eukaryote, which your Cyanobacteria clearly is not. They are not eukaryotes, they are prokaryotic algae. They are the only prokaryotic algae that we have. And this phylum, the phylum Cyanobacteria, the phylum Cyanobacteria, is pretty interesting in the sense that they are one of the major causes of algal bloom in your both freshwater and marine environment. These algal blooms usually, for the temperate regions, usually they occur mostly in the summer months or in eutrophic rivers, for example. So these algal bloom occur mostly on the surface of your ocean because remember, these cyanobacteria have gas vacuole so that they accumulate oxygen, and they only appear in the upper surface of your water because remember, these organisms are photosynthetic in nature. Okay, so before we start our lecture, let's have the general outline of our lecture for today. So first, we'll have the overview of our Cyanobacteria or your Cyanobacteria. Okay, so here we'll talk about their relation to other algae, their evolutionary history, and their basis of classification. So let's first go to their relation to other algae. So again, as I mentioned earlier, they are the only photosynthetic algae that we have. So, they are the only prokaryotic algae that we have. So aside from that, they also have what we call the chlorophyll A in your thylakoid. This is the first autapomorphic characters of the general algae character that we have. And since they are prokaryotic in nature, they have no membrane-bound organelles. So this is a very basic, knowledge for all of you that is in my class. So these are prokaryotic organisms. They have no membrane-bound organelle. And they also don't have any nucleus in that sense. Ayaw mag-click. Ayaw. So, in terms of evolutionary history, needless to say, we have been here for quite a while, and your Cyanobacteria is, the Cyanobacteria is the phylum that scientists think or a relative of your Cyanobacteria is the origin of your plastid. So it is the one that has been engulfed by your different primary plastid clades, including your red algae, your charophytes, and your chlorophytes, right? And also including those born out of your serial endosymbiosis and the secondary endosymbiotic event that we have. So yeah, the plastid, your Cyanobacteria is the origin of it's a relative of the Cyanobacteria. Our scientists think that it is the origin of your plastids. So in terms of evolutionary history, the chloroplast in these organisms can be traced back to your cyanobacteria in the sense. But separately, your cyanobacteria occurs in your bacteria or the eubacterial lineage. So they are included in that prokaryotic lineage right there in terms of its prokaryotic nature. And remember from our reading materials, these cyanobacteria has been eaten, or through the process of endosymbiosis, they have went into this green lineage and your algae lineage, and also through several endosymbiotic events to your heterocons and so on and so forth. So yeah, your cyanobacteria belongs to your eubacterial lineage, but then again, they jump or been engulfed through endosymbiotic event to your other algal lineage right here. And then serial endosymbiotic event, secondary endosymbiotic event to other algal, more complex algal lineage, your heterocons, your diatoms, for example, all those people. And your euglyphites, those organisms that we encountered in our first half of our course. Now, aside from this evolutionary history, so let's how do I close? Yo. So aside from that, we all know that your blue-green algae, your cyanobacteria, have been around for at least 2.7 billion years, according to our fossil records that we have. So scientists think that these organisms have been around at least the first 2.7 billion of, well, Earth is currently at 4.5 to 4.7 something billion years old, and your cyanobacteria has been, well, almost half of the existence of Earth, right? So yeah, they have been here for quite some time. Yeah, along with your other bacterial species. Yeah, and around 1.5 billion years ago, they can be considered the most dominant life form. We call this a point of in paleontology, and in geology, we call this the age of the cyanobacteria, because clearly your cyanobacteria, how do I delete your cyanobacteria? Oh. Nagiging O yung C ko. Ang hirap magiging arrow. Anyway, so your around 1.5 billion years ago, your cyanobacteria have been the dominant life form here on Earth, and scientists thinks that these organisms may have caused the first mass extinction that we know of, the Great Oxygenation Event. So let's call that the Great Oxygen. It's converting my or GOE. So yeah, scientists think that these cyanobacteria caused the Great Oxygenation Event back then. And the most important evidence for this is your the living fossils that we have in Australian coast, for example, your stromatolites. So your stromatolites looks something like this. So imagine something like this. Oh wow. not like that, but imagine it looking like this on your rock. So it looks like a rock, but it's made out of your minerals, your cyanobacteria, plus the mucilage that they secrete. And they look something like this because upon the years and years and years and years and years, many years of their life form, they stuck upon each other, and they form this column-like features in their form, in their this rock forms, these stromatolites. And you can actually measure the age by how many layers are formed all throughout those years. So yeah, scientists have predicted that these are very old living fossils off the coast of at the coast of Australia. And we are very fortunate that these stromatolites still exist, because remember, due to plate tectonics, your plates subsides down, they get melted and reorganized again and again through the process of plate tectonics. So it's a very rare that you get to have these living fossils from the time for a very long time that still exists. So it's it should be a national treasure. We should protect this as stromatolites. I mean, the Australian government should. But yeah, just imagine that these organisms, uh, for about 2.7 to 1.5 billion years ago, they existed here on Earth. They existed here even before plate tectonics. Just imagine that, even before the process of plate tectonics started. Because remember, back in the old days, nung wala pa talaga tayo, no? Remember that your mantle, back in 2.7 to 1.5 billion years ago, the mantle is a very hot, still very hot, and the convection current to for the convection current to happen, the mantle is still hot for that process to take place. So you have a convection current, but it's still limited on the it's very limited, because the mantle is still hot. So the convection current of the magma, no, they cannot sink down well. So it is still limited. With the motion of our plates back then, still limited in the shallower uh, portion of the crust, and the very thin crust in your mantle. Yeah, it's a very long time for these organisms to exist here on Earth. Yeah, and they form different symbiotic event that lead to the presence of chloroplast in almost plastid containing organism. No, so yeah, that's your the evolutionary history of your cyanobacterial lineage, creating the great oxygenation event, which caused the first mass extinction. They've been here for quite some time, and yeah, they experience a lot of history, no, in geological time scale. Yeah, those are your cyanobacteria. So in terms of their basis of classification, we can assume that most organism belonging to your phylum Cyanophyta lacks the organelle, your membrane-bound organelles. They have various photosynthetic pigments, including chlorophyll and various phycobiliproteins, which are important for maximizing their absorption spectra. And they also have what we call peripheral thylakoids. These are your chromoplasms, which again, if you remember, we have one thylakoid. Just ignore the arrows. I don't know what's wrong with this. It's making everything into an arrow. So one thylakoid per band. Remember that from our lecture. Oh, it has something to do with uh, my editor here. So yeah, so the thylakoids are the chloroplasts are located in the periphery, and you have ribosome scattered all throughout those those organism right here. So yeah, they also have a gelatinous coat and cell wall. They also have peptidoglycan layers right there. There are four orders here, different forms. Your let's move on to that. Your Croocales, your Oscillatoriales, your Nostocales, and your Stigonematales. So those are the four groups with different varying forms. Some has heterosis, if you remember your heterosis, and some are branching, some has false branching, some does not branch, some are colonies and unicellular. So those are the different groups that we have under your phylum Cyanobacteria. Now, in terms of their general characteristics, we'll talk about their cellular covering, their protoplasmic features, the gas vacuole pigments, and the growth metabolism. Let's move on to your the general characteristics. So your cyanobacteria, needless to say, has the simplest morphology among your algae groups. Simplest meaning most of them have a unicellular morphology, although some of them also have trichomes and your filaments. So the difference between your trichome and filaments, so imagine this figure right here. So this is considered a trichome because it doesn't have your sheet covering it. So that's a trichome. If it doesn't have a sheet, a mucilaginous sheet or a gelatinous sheet that covers the entirety of your filament of your cells, no, your continuous cells right here that forms your trichomes. Now, if it has sheet covering, that's now called a filament. So this is a trichome, but this is a filament. It's a filament if it has a mucilaginous sheet that covers it. Okay, now it can be multi-seriated, meaning one single layer of cells, or multi-seriated, no? Uniseriated, multi-seriated. When we say multi-seriated, it has multiple layers of your cells that comprises your filaments or your trichomes. So, but the main difference between a trichome and your filament is the presence of your gelatinous sheet. Other than that, your trichome and filament are basically the same, no? Yeah. So in terms of the main cellular covering of your cyanobacteria, you have, so imagine this from the inside going outside. So from the inside of your cyanobacteria, you have your cell membrane. Again, your cell membrane is present in the inside or the more interior portion. Again, your cell membrane is essentially just the plasma membrane. It's a layer that protects your organism and serves other function, of course. Aside from your cell membrane, they also have cell wall, which is this cell wall is, again, since this group is a cyanobacteria, they are a bacteria, they specifically are gram-negative bacteria, they have certain peptidoglycan covering. Oh, what? Huh? Peptido. Oh, Nagiging O yung C ko. Peptidoglycan. So yeah, so gram-negative bacteria, they have a peptidoglycan layer in their cell wall. They have, of course, a periplasmic space, an outer membrane, and your mucilaginous sheet. So aside from, or your capsule, your mucilaginous sheet or your extracellular polymer substance right there. So EPS or your capsule. Essentially, the function of your mucilaginous sheet, your capsule, and your EPS, the extracellular polymer substance, is for protection against desiccation. So again, your mucilaginous sheet and your capsule and EPS, they can hold much, they are hygroscopic in nature, they can hold much moisture and prevent desiccation of your organism. So that's the main function of your mucilaginous sheet. You also have your proteinaceous pili, which is composed of your pilin subunit, and this is, of course, responsible for locomotion, both the gliding and your twitching locomotion of your organism. So to take a look at what does this look like in your organism, just take a look at your synechocystis right here, showing your pili right here that it uses for locomotion. Also, you can see right here is your sex pilus that is mainly used for conjugation, no? Your exchange of genetic material for horizontal gene transfer and those things. And for the case of your oscillatoria, naman, we have gliding locomotion. So what's gliding locomotion? Essentially, there's no twitching in gliding locomotion, but essentially a certain form of foot processes is extruded by your organism in and out to glide across surfaces. So yeah, or sometimes they produce sticky substances to stick and pull themselves across the surface of whatever they're moving across. So those are your oscillatoria and synechocystis. Moving on with your protoplasmic features, we have your thylakoid right here, which are in terms of location, they are located in the periphery of your in the periphery of your organism, of the near the cell wall of your organism or the cell membrane of your organism. They have no granum. Again, these are one thylakoid per band. There are no stacks of thylakoids. The pigments are, of course, your chlorophyll and certain phycobiliproteins. And your main storage product, as you remember, they have glycogen, and which is unique to your cyanobacteria, are your myxopycian starch. So your myxopycian starch. Ang daming arrow, no? Huwag niyo nang pansinin yung mga arrow na yan. Anyway, so that's your myxopycian starch. I use green kasi blue-green algae sila. So yeah, your myxopycian starch are the main storage product aside from glycogen. Again, myxopycian starch are unique for your cyanobacteria. They also have scattered ribosomes, no, across their body, I mean their cell. You also have a circular DNA called a nucleoplasm in that. And you also have different proteins or granules that you can observe. You have cyanophycin, which are granules of your which serves as temporary nitrogen reserve of your cell. Again, nitrogen is very important for protein synthesis later on the life cycle of your organism. You have carboxysomes, which is usually composed of your carbonic anhydrase and rubisco. Again, rubisco is important for fixing carbon dioxide with RUBP, and your carbonic anhydrase naman, kung maalala nyo if you remember in your plant physiology, carbonic anhydrase, or in your microbiology, carbonic anhydrase serves the purpose of producing CO2 from bicarbonates. So usually for, again, these are marine organisms. They utilize bicarbonates to produce carbon dioxide sometimes. Okay, and we also have different polyphosphate bodies and when you took your bio 120, you remember your polyphosphate bodies are metachromatic, or these volutin granules serves as your phosphate storage. Again, phosphate storage are needed for your DNA and your ATP, the phosphate and the pyrophosphate, yung mga ganyan, di ba? In your ATP and in your DNA too, the backbone of your DNA. And you also have, polyphosphate body serves as phosphorus storage. You also have polyglucan granules, which contain CHO or your sugars. So these mainly serve as an energy storage of your organism. Polyglucan granules. Okay, so yeah, so you can see here the one thylakoid per band. You can see it in the periphery of your organism right here. Very organized structure and different components of the cells inside of it. Okay, so moving on with your gas vacuoles. Your gas vacuoles is, of course, these are comprised of gas vesicles, or these hollow cylindrical-like tubes. They sometimes resemble honeycomb-like structure to maximize the tessellation in the gas vesicles, in the gas vacuoles. But essentially, again, if you remember in our Bio 120 class, these gas vacuoles has certain they have beta pleated sheets and alpha helical components, and these are used to exhibit a certain permeability to gas, certain degree of permeability to gas, but it exclude water molecules. So yeah, it only allows air molecule to enter, thus creating a positive buoyancy force, positive buoyancy for your organism, which again is important for, again, sunlight, exposure to sunlight, because these are photosynthetic organisms. So in terms of pigments, they have chlorophyll A and B, and some organisms like your Acaryochlorus has a chlorophyll D too. You also have different carotenoids, including your echionine, which is a carotene-based carotenoids, which are carotenes. You also have mixoxanthophyls, which is a type of xanthophyll, yellowish to blue, yellowish tinge of color. You also have different phycobilic proteins, your phycocyanin, allophycocyanin, phycoerythrin, phycoerythrocyanin. These are your different phycobilic proteins that, if you remember from our previous half of our semester, these are used in maximizing, well, it became a mirror. These are used in maximizing the absorption spectra of your, maximizing the wavelength that the light wavelength that this organism can capture. Because some of them are marine organisms. But yeah, so in terms of photosynthesis, the main energy source is, of course, sunlight, but the main electron source is your water molecule. But in certain cases where there is absence of water or absence of sunlight, some organism can turn to hydrogen sulfide, as a source of electron if water is absent or there's no sunlight, for example. Or sometimes they can use that as a source of energy too. Some representative, I mean. So in terms of growth and metabolism, there are three different general nutritional types here. We have your obligate photoautotrophs, meaning just like plants, they are obligate photoautotrophs, which captures the energy of your sun and synthesize organic molecules, carbon molecules from inorganic sources, from carbon dioxide in the atmosphere. Yeah, and some are photoheterotroph, meaning they also use the sunlight as an energy source, but instead of inorganic carbon like carbon dioxide, they use organic sources like your sugar molecules that are already been synthesized, or other organisms, other parts of organisms. No, they consume their carbon, they consume their organic carbon from other organism. They don't capture the carbon dioxide in the atmosphere. So organic carbon yung kinakain nila, yung source ng carbon dioxide yung mga yan. We also have facultative chemoheterotrophs, which can change, no, their preference from obligative photoautotropic to chemoheterotropic mode of life cycle or nutrition. If they are put in certain conditions where sunlight is absent. So those are your nutritional type. And moving on with the symbiosis, we have two types here. We have extracellular association and intracellular association. So let's talk about those two. So when it comes to symbiotic association or interaction, you need to always talk about your cyanobacteria. They are very important in terms of symbiotic interaction. First of all, let's talk about the extracellular association, the living together, just together, not inside each other. So living together, okay? These are extracellular association of your cyanobacteria. And usually, this type of growth is very slow. They have very slow growth. They usually perform to assimilate carbon dioxide and usually ammonia or ammonium or nitrogen together. And there's a lot of heterosis involved in this type of association. Examples, famous examples are your nostoc and your azolla. Your nostoc, ito na yung, this one we have already encountered in our laboratory portion. This one forms actinides and your heterosis, those circular anoxic environment for your cell, diba? Kung maalala nyo. So those are your actinides or heterocysts. These are your nostoc or anabena, no, nostocales in general. Your azolla, these are mosquito ferns, or sometimes they are generally they are the aquatic ferns that we have. So in terms of their extracellular symbiosis, your azolla, your aquatic ferns, provide shelter, while your nostoc provides nitrogen storage for your mosquito fern or your aquatic fern. So that's how they benefit from each other. While your more famous one, again, maybe you encountered this in your city, in Baguio, there are a lot of lichens here. So siguro next time we can accommodate another portion exclusively for lichens, and how your algae is in close association with your fungi. So your lichen from your basic biology course is a symbiotic association of at least two, but there are this paper that says that there are at least 3 organisms involved. But for now, let's just say there are 2 organisms that are involved in this symbiotic association. We'll have your algae, the pycobiont, and your mycobiont, the fungi. So the purpose of the fungi is to house or uplift the organism from the soil So usually your fungi will provide the housing and will attach your organism to the branches of the tree that is way beyond your soil, diba? Nasa ano siya, epiphytic in your high branches of your tree. And so in a way, your fungi houses and uplift the organism, the entire assemblage of your organism in this above the soil and to gather more sunlight. Yeah, so that's the purpose of your microbiome, the fungi. So basically the most uh, um, fungi groups that we have here that host certain microbiomes. So your microbiomes usually belongs to the group of ascomycetes. Sorry, that's my oh oh ascomycetes, and from your group Basidio. It's a struggle to write here. Basidiomycetes. So ascomycetes and basidiomycetes. These are the general microbiomes group that we have in your lichen. Your phycobiont, on the other hand, provides the food. I mean, any excess food that your phycobiont produce, it gives to the microbiome. So that's how this symbiotic association between your phycobiont and microbiont goes together. Moving on with your intracellular association, these are the literally living within each other. We have your cyanelle and your cyanome. Your cyanelle, these are the in a way you can think about it as the origin of your organelle, and your cyanome, on the other hand, is the host. So cyanelle is the origin of the organelle, cyanelle or organelle, and your cyanome is the host. And this is, needless to say, this is the most famous example of intracellular association that we have, that eventually came into the development of your chloroplast across different groups, across different phylum that contain the plastids that we have. And another example that we can issue here is for the case of your Montastria cavernosa. This is a coral species which has species of coccoid cyanobacteria inside them that enables them to photosynthesize. So these are kind of like your zooxanthellae, but of course zooxanthellae are a a golden brown or brown algae representative. Yeah, but for the case of Montastria cavernosa, it has this coccoid cyanobacteria inside them that allows them to photosynthesize. Okay, so now moving on to second to the last portion, we'll now move on to the morphological diversity and ecology of your organism. We'll talk about different adaptive forms, the reproduction process and ecology of your cyanobacteria. Oops. Let's talk about first the most common and the simplest forms that we have here, the unicellular or the colonial forms. Again, your colonial forms are just aggregates of your cells, no? The unicells, but these aggregations are formed due to mucilaginous secretions or due to, well, them sticking to each other. So most of the unicellular and the colonial forms are phytoplanktons of the world. So these are the primary producers in most ecosystem, in most aquatic ecosystem, especially in most aquatic ecosystem. And they produce the much needed the much needed oxygen that Earth, us heterotrophic organism needs. Examples of your phytoplanktons, unicellular forms of your cyanobacteria, we have your prochloron, which are, so these prochlorons are these organisms which are endosymbionts of this tunicate species right here that usually clings on your bedrock or some certain rock formation in your marine sediments. I mean, marine rocks in littoral areas. So those are your tunicate endosymbionts. Sometimes these are called sea squirts. So in the transition, if you remember in your, in Zoo 111 and Zoo 102, so in vertebrate zoology and in in comparative anatomy of your vertebrates, your tunicates are closely associated. Well, they are invertebrates, but they are closely associated with your chordates, diba? If you remember from your classes back then. So yeah, that's your prochloron. You also have your prochlorococcus, which are these bright fluorescent organisms right here. We also have your microcystis. These microcystis, on the other hand, are also responsible for algal blooms that we have. So these are your microcystis right here, colonial in form. So aside from your unicellular and colonial, we also have filamentous forms of your organism. These filamentous forms, you have your different types. You have branch or unbranch. Meron pa tayong tinatawag na false branching, true branching, we'll talk about. And we also have uniseriate and multiseriate type of organism. So let's look at your, well, when we talk about uniseriate, you only have one layer, okay? One layer of cells. Pangit ang drawing ni sir. So one layer of cell, your multiseriate can look like something like this. And sometimes it can have mucilaginous sheets covering them. So yeah, looks something like that. Okay, multiseriate, uniseriate. You have branch. So branching, let's draw the branching, or just simple unbranch filamentous algae, like that. Now, let's talk about more and review. Again, let's review the process of the figures of your branching and false branching, true branching, something like that. Again, this is just a recap of your laboratory portion. The false branching right here, as you can see here, the mucilaginous sheet breaks apart in this area. So it's not continuous. So you can see here, this is for your Cyanotonema species. This is Cyanotonema type of branching. These are clearly for false branching. And this type naman of false branching where a heterosis or some actinids form in this portion right here, and there's no continuation of your cell division right here, right? These are your Tolipotrix or your Calotrix species, if you remember in your laboratory. These are those type of false branching. So yeah, there's no continuous sheets right there. But the true branching right here are examples of your Stegonema. Let's just write them. Stegonema. These are Tolipotrix. And yung mas famous na ginamit natin sa lab, Calotrix. And these are your Cytonema. Cytonema. So yeah, so those are just a recap of the laboratory portion that we have. Don't forget them. That's what I'm saying. Next is we need to talk about the heterosis. This is very important, especially in this phylum, your Cyanobacteria. So your heterosis is not this, but this. So your heterosis, these are large and thick-walled, well, very anoxic environment, meaning there's no oxygen, and usually you can see right through. It's very clear. Unlike your actinids, which has certain granules inside them, your heterosis So very clear inside. It's very thick walled. As you can see here, there's a lot of layers of wall right here that prevents the presence of oxygen inside that specialized cell. So why is it like that? Because, well, first of all, it's a site for end fixation. Why is it anoxic? Because there's a need for oxygen to there's a need for that area to be devolved of oxygen, because remember, these heterocysts are a site of end fixation, and nitrogenases are especially fecal enzyme. Nitrogenase. So your nitrogenases are a very fickle enzyme in the sense that they are very sensitive to oxygen. When there's oxygen around, they get to be destroyed, or they get to be they change conformation, something like that. But yeah, that's why it's devolved of oxygen. Aside from that, your these heterocysts also comes from your vegetative cells. So what happened is your vegetative cells first they lost the carbon or the sugars inside them. So what happens is, in the development of heterocysts, your cell will lose its CHOs, or its sugars. So it will deplete its sugar content inside, and it will become what we call the proheterocyst. This portion of the development of your vegetative cell, when it loses its sugar molecule, sugar reserves inside them, this proheterocyst is still reversible. But once the cell thickens, once the cell thickens their cell wall, there's a thickening of their cell wall, and their thylakoids are now gone, meaning thylakoids are dead or something like that, and they lost this thylakoid, and the walls are getting thickened. When you see a thickening of the cell wall already, that means the process is irreversible, cannot go back to its old state. It will only become a heterocyst, because again, if you have thick cell wall, you have no oxygen, and oxygen is indeed required for cellular respiration, remember? So once you have a thickening of cell wall and your thylakoids are gone, and you produce nitrogenase enzymes, the process is now irreversible, and the cell will only become a heterocyst. Again, let's review from a vegetative cell. Your organism will deplete or lost their CHO, or their sugar reserve, and they will become a proheterocyst. After that process is still reversible. After that, they will lose their thylakoid, they will produce nitrogenase, they will have a thickening of cell wall. That process, that second process, is now irreversible, and they will now become your heterocyst. So yeah, those are the event that happens, and at the very end, the thickening of the cell wall, you'll have a terminal event called your apoptosis, which is technically the organism will be devoid of life, kind of like that, no? In which it will only serve a purpose of nitrogen fixation. So essentially, after the terminal event, apoptosis, the cell will now become photosynthetically inactive, in which your microplasmodesmata will be the only choice of food, no? So your microplasmodesmata here will be the only site where CHOs, or your sugar molecules, or food is delivered by the adjacent cells through that microplasmodesmata right there. So that's the only site of your delivery of food. No, because again, the cell, the heterosis, sorry, not this one pala. No, tinuturo natin yung actinides kasi malaki. This one. Okay? That's not this one. These are actinides. So that's the only site of entry of food molecule from your organism. Move. Okay, so as nag-move din siya. So, what chemical process happens inside your cell? Well, first of all, you have your nitrogenous enzyme, and in the presence of metal catalase, metal catalytic enzymes. In the presence of your metal that acts as catalytic agent in this reaction, you will need ATP or energy molecule plus your nitrogen N2 in your atmospheric N2 in your atmosphere. You will create the most important here is these two molecules of ammonium cation. So essentially what happens here is nitrogen fixation. So again, nitrogenase plus your metal cation, sorry, it's just hindi siya gumagalaw. So yeah, we have where are we? Nitrogenase, the enzyme, the main enzyme. We have your metals that will act as catalyst of your reaction. We have atmospheric N2 and a lot of molecule of ATP, a lot of energy to convert your atmospheric N2 to a much more amicable split. Amicable, a much more usable form of nitrogen. No? We have your ammonium cations. Mga kamag-anak na yan ni ammonia. Your ammonium cation will then go to another process. Again, if you remember, we have your citric acid cycle right here. In the citric acid cycle, we have 2-oxoglutarate, and in the cycle, no? Diba, maalala nyo, may part dyan na 2-oxoglutarate, and that 2-oxoglutarate can be involved in this process wherein your ammonium cations can be incorporated to your amino acids to the presence of your glutamate dehydrogenase, in which essentially what happens is ammonium cations or nitrogen molecules is fused into your amino acids right here, producing now your glutamate plus water. Your glutamate can then, so punta tayo dito, glutamate can also be transformed into glutamine, which has 2 molecules of, ayan. So yeah, that's your nitrogen cycle involving your nitrogen fixation involving your heterosis. Yeah, there's a lot of energy that, why is it important for the organism to do this? Well, if you remember, nitrogen is a very important and a very limiting material for life in a marine environment. No? And for basically nitrogen, there's a lot of nitrogen in our atmosphere, but the form of nitrogen in our atmosphere, 70% of atmospheric nitrogen is inert. So in that sense, we cannot use nitrogen in our atmosphere. That's why your organism needs to fix that nitrogen, convert it to a more amiable material or manageable material for the cell to use in its catabolic or anabolic activity, in its metabolism, in protein synthesis, for example. Yeah. So yeah, That's how your ammonium cations are incorporated to 2-oxoglutarate and to your glutamate to form your glutamine. So glutamate and glutamine, these are nitrogen carrying amino acids, remember? So yeah, those are your techniques of your organism that it uses. Yeah. Now, you also have, so aside from heterosis, in terms of reproduction, we have your actinids. Your actinids are the first one that we talked about, diba? These are the very large cells that forms. But just like your heterocyst, just like heterocyst that forms, well, your heterocyst forms during times of a very limiting nitrogen environment. If you don't have nitrogen, your organism will most likely form heterocyst, these heterocysts. Just like your heterocyst, actinids also forms when there's a very harsh environment, when the cell feels that it is in a harsh environment, or it's in a certain condition where there's no water, or certain stimulus that causes it to create these actinids. What will happen is that your organism, that is your vegetative cell, so your normal vegetative cell, will stuff itself of this, stuff its protoplasm of different sugar molecules and different storage granules inside it. It will stuff the cell with different granules, and this conspicuous granulation is of your glycogen storage and your cyanophycin storage pigments. These are essentially food that are being stuffed inside that vegetative cell in the first place, in the protoplasm of that vegetative cell. And what happens in that actinid is that when it forms, it is usually enable the organism to withstand extreme environmental condition. So extreme cold, extreme environment, the actinids can survive that. So it more or less acts like a, if you remember in your Bio 120, we have the endospore. It kind of acts like that. But unlike your endospores, your actinids will germinate. No, it's like a seed that will germinate into a new filamentous algae after the condition went back to normal. Okay, yeah, kind of like endospore too in that sense. So that's your actinids. Yeah, ito nyo, nag-grow siya. So in the Avon presentation that we have, the second picture of the table of contents right there, those are the, well, we agree if it's not a spore, no, the picture in the table of contents in the Avon presentation, if it's not a spore, it's an actinid, because it looks like a germinating actinid. So why we use that as a background right there. So yeah, those are your actinids that can be used for reproduction right here. Aside from that, your organism can also undergo asexual reproduction by means of fragmentation. Let's just write that. Oh my God. Why? Nako. Nako. Fragmentation or cellular bisection. Cellular by God. No. I hate annotating PDF. So yeah, fragmentation or cellular bisection. So there are two types. We have your hormogonia or your hormogones and your bayocytes or the endospores. So your hormogonia, these are produced essentially by your filamentous algae. They are detachment of short pieces of trichomes without your mucilaginous sheath, and they exhibit a positive phototaxis or gliding to buoyant nature. So what will happen is a certain part of your trichome of your filament will be detached and it will be ejected and it will rise to the surface to photosynthesize. So that's your hormogonia or your hormogones. Your bayocytes or your endospore, on the other hand, these are usually produced by your cocoid species by several division of your protoplasms. So you can see right here. And these are usually ejected or released via the apical pores or the apical portion of your organism, the apical pore of your organism. So your bayocytes, your endospore, are essentially smaller daughter cells, no, or similar, no, asexual kasi, similar copies of your daughter cells. So those are your bayocytes and your hormogones. Move. Now, moving on to the last portion of our lecture, we have your ecology of your organism. So in terms of ecology, they your cyanobacteria occupy a very wide range of environment, but most of this they perform one major role in the ecosystem, in the aquatic environment. They are the primary producers in most environments. So in marine habitats, for example, in your littoral zone or your intertidal zone, yung malapit dyan sa dagat, no? Yung sa dalampasigan, I mean, they form this encrusting film dun sa ating mga littoral, dun sa mga rocks. Oh, wow. Wow. Yung siyang cloud. Dun sa ating mga rocks. What? So, yan sila. So they form this encrusting film in this portion. Most of them can fix nitrogen and they contribute to the primary productivity in that area. So mga primary producers talaga to. In open oceans, they are also primary producers in open ocean, but certain tiny cocoid species can autofluoresce, no? And they can light up, no? Alongside with your diatoms too. Sila yung mga umiilaw dyan. Those are your phycophytoplankton, which are more smaller phytoplanktons. In brackish water, they can also be found in brackish water in estuarine environment. Also, in freshwater ecosystem, they are present. We also have psychrophilic and psychotropic species. When we say psychrophilic species, these are algae species that really loves this cold environment, meaning you can find this in the Arctic and Antarctic portion, yung malalamig na portion of our world, of the globe. Your psychotropes, these are more present in the temperate areas of our of the earth. Oh my god, temperate. Temperate. You know what I mean? Pagpasensya nyo na yung sulat. So, temperate regions, these are just tolerant to winter, but they don't necessarily love the cold. But your psychrophiles, those are your sadistic individuals which love cold, just like Mother Russia. So, yeah. We also have thermophilic species, which can be found in non-acidic hot springs. We also have a representative that can be found in terrestrial environment, for example, your lichens, diba? And also in the soil. And when they are found in the soil, they can improve soil quality, they can improve the binding and reduce the amount of soil erosion. They can increase the soil moisture of the soil moisture, since they form these mucilaginous secretions. They can also fix nitrogen, which most of the time is used by your plants, diba? So hindi lang mga fungi yung kaya mag-fix, diba? Yung mga rhizobium na sikat ng nitrogen, sila din. They can also be found in an hydrobiotic environment, like for example your nostocommunes, which are, when we say an hydrobiotic environment, these are environment with no to a very little water concentration, water content, and these nostocommune can still survive even without, or even with a very little water in the environment. We also have your cryptobiotic crust, yung tinatawag natin mga desert glue, just like your anhydrobiotic, but this time these can be found in your algal blooms. These are called the algal blooms, yung mga desert glue na yan. These are algal blooms in your desert or dessert. Yung matatamis. Hindi yan. Joke lang ni sir. Now, moving on to the last portion, we have your parade of taxa. We have, again, as we mentioned earlier, we have 4 taxa involved here. We have your Croococales. These are unicells and form mucilaginous secretion to form your colonies. We also have your Osciliatoriales, which are filamentous in nature. So we have your oscillatorial right here in letter C. We have your Prochlorococcus in your Croococales. And your oscillatoriales are filamentous, but they don't exhibit branching, and they don't form heterosis, and they don't form actinids. Your nostocales, on the other hand, these are yung mga na-mention natin na anabaena, nostoc, yung mga yan. Ito yung mga filamentous and branching na mga organism. They are filamentous and branching. And they are the ones that form your heterosis right here. And wala kang makitang actinids. But yeah, ayun, natatakpan. Ay, hindi, bukang cell pa din yan. Not sure. But yeah, actinids are larger naman. So yung makikita nyo. And your Stigonematales, on the other hand, these are, for example, here, your Hapalocypon. These are filamentous. They are true branching na mga species, and they don't form your heterosis and your actinids. So those are the major taxa that we have here. You can have another entire course just to describe these species of these groups under your cyanobacteria, your Croococales, Osciliatoriales, Nostocales, and Stigonematales. But I hope by providing these examples right here, you can experience the diversity within this group of organisms. Thank you guys for tuning in to our lecture for today. And if you have any questions, don't forget to chat me or message me through my email address. And that's all for today. And thank you for watching. This is Sir Patrick. Peace out.